All right, welcome back. So in this video, I want to talk about a test cross. And first, I will explain to you what it is. And then we're going to see how Thomas Hunt Morgan used the test cross, looking at his wide-eyed mutations. All right, so let's say, for sake of argument, that I'll give you a green pea. And I say, what is the phenotype of this green pea? And you say, well, it looks green to me, so the phenotype is green. And I say, what is the genotype for it? And you said, green, I remember, is recessive, so it must be 2 lowercase y's right here, okay? And if you said that, then you would be correct, okay? And for sake of argument, now I am going to give you a yellow pea, okay? And I will say, what is the phenotype? And you say yellow. And if I say, what's the genotype? You simply don't know. It could be that it has 2 capital Y's, or it could be that's supposed to be R, or it could have a capital Y and a lowercase y. And the question is, how do you figure it out? And the answer seems remarkably simple. If you want to figure out whether this one is homozygous dominant or heterozygous, all you do is you cross it with the recessive, with the homozygous recessive individual, okay? That's called a test cross. A test cross is if you don't know what the genotype of a dominant individual is, you can test cross it to the homozygous recessive individual. And now, if all the offspring is yellow, then you knew it had to be this, because then every offspring receives one of these capital alleles, okay? If the offspring is a 1-to-1 ratio of yellow versus green, then you knew it had to have been like this, where everybody receives the capital 1 is going to be yellow, and everybody who receives the lowercase 1 is going to be green. Why? Because this one right here can only give you the recessive allele, and whatever the dominant is going to mask that. So that's what a test cross is, okay? And we do it because we don't know sometimes what the genotype of this individual is. And that's exactly how that's exactly what was used by Thomas Hunt Morgan. I'm going to give myself a pointer again. And so recall, please, that in his cross from last time, all the females had red eyes, right? And he said, I can explain that because all the females have received an X chromosome with the normal wild type allele on there, right? This, okay? But some people said, or alternatively, what if the white allele is lethal in females? So what if you don't see white-eyed females because they get killed, they don't develop? For some odd reason, the white-eyed gene or the white-eyed mutation kills all the females, but the males live. That would be an alternate explanation why every single female had red eyes, right? Okay, and Thomas Hunt Morgan said, you want to see a white-eyed female? I will show you a white-eyed female, okay? And he did so because he knew that this F1 hybrid right here, okay? So that is the first generation. We take a white-eyed male and a homozygous dominant, well, wild type, red-eyed female. And in the first generation, you get a hybrid. That's my F1, right? So she basically, so let me write this down quickly. She has one X chromosome that is W plus, and the other X chromosome that has the W on it. Now, she's perfectly red-eyed because, again, this XW plus right here is the red-eyed dominant allele, and the mutant right here is recessive, okay? But we can do a test cross to actually show this to be true. Okay, and that's where the test cross comes in, right? What is a test cross? A test cross means a cross to a mutant, to a recessive individual. Well, since the white mutation is recessive, this would be the one. This is also the dad, by the way. so this time we're not doing brother-sister matings. This time we're doing daughter-dad matings. Even worse, again, in fruit flies, perfectly okay. Okay, but it's a test cross because we're crossing an individual that has unknown. I can't look at her and say, oh yeah, she must have one red and one white-eyed X chromosome, but I'm going to figure it out by crossing it to this male right here, which is hemizygous for the white-eyed mutation. Okay, now that we can figure out our table right here, she produces two kinds of gametes. Half of her gametes are going to have the red-eyed X chromosome, half the gametes are going to have the white-eyed one. And this male is going to make two kinds of gametes. Half of them are going to have the white-eyed X chromosome, the other one are going to have the Y. Okay, and so if we figure out our table right here, right, so this one is right here, this one is right here, and then these are the ones from the female, what we see is that we get four different kind of fruit flies. This one right here is a female, 2 X chromosomes, and one of them has the red, has the wild type red gene. So that allele, that means that this is a red-eyed female. This female right here has 2 copies of the white mutant, recessive mutant, one from dad and one from the mom. And she would be a white-eyed female if in fact white-eyed females are not lethal, okay? And then here we have 2 males, one receives the X, because males always get their X chromosomes from their mom. Half of them are gonna have the red, half of them are going to have the white, and dad furnishes the Y chromosome right here, okay? And so we have 4 different kind of fruit flies: red-eyed female, white-eyed female, red-eyed male, white-eyed male, okay? Red-eyed female, white-eyed female, red-eyed male, white-eyed male at equal proportion, and that means that the white is not lethal in females, because check it out. Here we have a perfectly white-eyed female. And that's a test cross. We have now unearthed the fact that this was actually a heterozygous individual versus a homozygous one. Okay, so that's how the test cross is used right here. So in order, as part of this work, he performed this test cross. So that's my heterozygous female mated with a white-eyed male. The other cross that was performed was a reciprocal cross, okay? So now that we have a white-eyed female, right? So this is a white-eyed female, XW, XW, 2 recessive white-eyed mutations on the X chromosome. We can cross it with a red-eyed male, okay? Red-eyed male, white-eyed female. And let's see what happens, okay? Well, the dad gives the Y chromosome, and every single one of the offspring that is male is going to have an XW. Okay, so all the dad was white-eyed, but all the sons are dad was red-eyed, but all the sons are going to be white-eyed. Okay, the dad is going to give this XW plus to each and every single one of his daughters, and right there we can stop, because every daughter is going to be red-eyed, because they're going to have the XW plus, the red-eyed wild type allele from dad. So it's really, it's called a reciprocal cross, because we're switching. We're starting with a red-eyed male and a white-eyed female, and what we get is red-eyed females and white-eyed males in the next generation. So again, because Thomas Hunt Morgan now understood how these sex chromosomes work, he could make a prediction about the outcome of his crosses. And when that prediction came true, this became real science and was publishable. And so that's Thomas Hunt Morgan's claim to fame, all resting on these sex-linked traits, specifically the white-eyed allele. But of course, he did it with many other mutations on these chromosomes, and ended up mapping these chromosomes, which we'll talk about in a later module. Okay, we'll be right back.